behind your sternum in front of your heart. For people taking the lab practical, if you see a gland right here, that's the thymus gland. By the time you're an elderly individual, you might not even have a thymus gland anymore. It might be totally replaced by fat and scar tissue. So it peaks out early in life, secretes a bunch of hormones, including thymosin. So I would recognize thymosin as the primary hormone. And the T in T cells or T lymphocytes comes from the fact that early in their life, where are all blood cells coming from is after you're born? All blood cells are made in red bone marrow. So they're all coming from bone marrow, but early T cells migrate to the thymus gland and mature and get programmed in the thymus. 99% of early stage T cells never make it to your circulation. They get weeded out because the last thing in the world you want are T cells that don't work right, as in like lupus and stuff, the autoimmune diseases where the T cells are attacking your own tissues and whatnot. So thymosin is a programmer to make sure T cells work right. So this happens early in life. A couple hormones that are making a lot of news. Leptin is produced by adipocytes, fat cells. And it's called the feel-full hormone, satiety, which is a hard word to say, but that just means feel full. So this, you have receptors in your hypothalamus, which is your appetite center for this hormone. And when you eat fat, you release this hormone, and it makes you feel full. And it triggers, believe it or not, when you eat, the reason you lose weight when you eat fat is because of this hormone. When you eat fat, you release leptin, and leptin triggers use of fatty acids to make energy. Leptin, some people now are saying, is the major influence of the thyroid hormones. More powerful than T3 and T4, which historically T3 and T4 have always been thought to be the major metabolic stimulators. But this is why you need to eat fat, or one of the reasons, besides some of the basic nutrients in fat. When you eat fat, you feel full. It suppresses your appetite. And when you eat fat, you burn fat for energy. A high sugar diet inhibits leptin and stimulates ghrelin, which is the hunger hormone produced by your stomach. But it also has receptors in the hypothalamus, which again is the hunger center in your brain. So these two hormones are antagonistic, and if you read up on leptin, a lot of people, high sugar diets can lead to leptin sensitivity problems, where now you can't burn fat at all because you're locked into this situation. If you eat a lot of sugar, what's that excess sugar going to get turned into? A lot of fat. So you start loading up your adipocytes with fat, and you produce too much leptin, and then your body just stops responding to it. And you get locked into a vicious cycle of leptin insensitivity, and you can't lose weight. Your body is just not burning fats for energy. So this is a real important hormone.
Would chocolate be considered fat or sugar?
Chocolate is neither.
Oh.
They add sugar to chocolate to make it more appeasing, but chocolate comes from cacao beans, and I don't know what chemical class chocolate would be in. It's not. The carbs are added to make chocolate sweet.
All right.
And chocolate by itself, as you've heard, dark chocolate is actually really beneficial. It's got a lot of antioxidants and all that kind of stuff. So good for you. The darker it is, the better. But anyway, leptin makes you feel full, and fat cells make it. And if you eat fat, you'll release more leptin, and you feel full. Ghrelin, just the opposite. It makes you feel hungry. Sugar stimulates more ghrelin to be released. You ever notice if you're eating a bag of chips, you can't stop sometimes? You just don't get full, and the whole damn bag is gone before you know it. But if you're eating like a fatty meal, you get full real fast. You know, you taste good, but you just got to stop eating. You're full. It's all about hormones. You want to lose weight, learn to manipulate your hormones. That's what dictates whether you store fat or burn fat. And just throwing this in there, there's a medical doctor who's written a pretty interesting book. His last name is Johnson, and it's called The Fat Switch. highly recommended book. You can get it on Amazon. And he talks about, of all the different sugars out there, high fructose corn syrup, and how bad it actually is for you. But definitely, because it's stimulating ghrelin and inhibiting leptin, triggers fat gain, and is the most fattening substance on the planet. Interesting book to say the least. But just summing it up, leptin makes you feel full, ghrelin makes you feel hunger, and shows you what stimulates each one and what inhibits them. So we'll fly through these. And real quick, your textbook now talks about, there's a guy named, a German medical doctor named Hans Seely, who people call the father of stress, because he kind of came up with these 3 stages of the stress response of the body. And first of all, some stress is good. Like moderate exercise is a stress, right? But it's really good for you. Moderate exercise increases insulin sensitivity. So if you're exercising 5 days a week, you're going to respond better to insulin. Just one of millions of examples. And that's called eustress. Distress is where you have too much stress and you overwhelm your body's stress mechanisms. So we're normally subjected to stress and we deal with it. And that's adrenaline hormones and cortisol. And this guy calls it the general adaptive syndrome, or GAS, the sequence of your body's, how you deal with stress. And there it is. You get the initial stage is your adrenaline response. Got to come up with a lot of energy real fast to deal with the stress. But you get scared. As an example, famous study done by psychologists, and they took not good to be a rabbit in terms of studies. They took these rabbits and they put them in little tiny cages, and they put the cages inside a cage of a tiger. And the rabbits fail to thrive. The tiger can't get at them, but they die real quick. It's because they go through this real fast. They're constantly secreting cortisol, and they exhaust their adrenals, and when they get to stage 3, they die. So that's how important controlling stress is. Initial stage should be short term. Your adrenaline hormones are quick fix hormones for stress. So they're the short term stress hormones. If the stress doesn't go away, now you got to start hyper-secreting cortisol. So cortisol is the body's long-term stress hormone. Stress comes, and this guy talks about it's not just emotional stress, chemical stress, electromagnetic stressors, physical stressors. Too much exercise is a stress. So if you over-exercise, a lot of guys in the NFL and baseball and stuff like that, that's overkill. Exercise is good, but those guys wear their bodies out. Football players don't live that long. They die 15 years younger than the rest of the population. Part of it is all the massive training, and obviously the physical beating they take is part of it as well. So the resistant stage is cortisol, hypersecretion of cortisol. Eventually, the gland wears out, and when the adrenals wear out, that's when really bad things happen. Your body starts to literally fall apart. It's kind of interesting. Cortisol, they talk about, as one of the major inflammatory regulators of the body. Too much cortisol, real bad, but too little cortisol, even worse. If you're not producing cortisol at all, your body's in a never-ending state of on fire, inflammation. And again, bad things happen, heart attacks, you get cancer, blah, blah, blah. And this is just cortisol secretions throughout the day. Like all hormones, you secrete hormones in spurts. And if you're getting up early in the day, that should be your peak cortisol secretion. It mobilizes blood sugar and you make energy and all that. And it goes up and down throughout the day. And then by nighttime, should crash. Cortisol levels should be real, real low. But if it's 11 o'clock at night and you're watching the late night movies or whatever, and got the light of a TV 5 inches from your face, and then you go to sleep and your electric face clock is 6 inches from your head and all that, your cortisol levels are still massively high. And again, they talk about this being epidemic proportions. And again, cortisol out of whack, real bad problems. Elevated cortisol is famous for, you're trying to come up with blood sugar. Cortisol is a gluconeogenesis hormone, especially for muscle tissue. So whereas glucagon is also a gluconeogenesis hormone, it doesn't work on proteins like cortisol does. This is where cortisol gets a bad rep. You're going to break down particularly muscle mass in your extremities and take those amino acids and turn them into glucose to support the stress response. And then any excess glucose not used gets redeposited as fat, primarily in the trunk and the core of the body. So hypercortisolemia is associated with a pear-shaped body. And if you look around and you see people with skinny arms and legs, but that midsection, or even people that exercise that can't get rid of this, it's hormones, and they're probably hyper-secreting cortisol, and it doesn't matter how you go to the gym and you get on the treadmill for an hour, you're just elevating your cortisol levels even more because your body's not coping with stress at that point. So again, it's ultimately about hormones.
So how would you change that? Like, how would you stop like if you were trying to
If you're hyper-secreting and you want to,
yeah,
that's a tough battle. You got to reduce stress. One of the big triggers for cortisol is toxicity. If your body and you're overwhelming your liver with fatty toxins and things like that, you're going to get a stress response. So eliminating toxicity can dampen the cortisol response. That's one possibility. Serine is remember Focus Factor on television? They used to advertise that brain formula. All the brain formulas have an amino acid derivative called serine in it. And some people consider that like you can use that and it will actually modulate, control your cortisol response. So a lot of people that have insomnia and stuff like that, because they have a high cortisol response late at night, will take that and it calms down the cortisol response at night. That's one possibility. And get rid of stress. How you do that, I haven't figured that one out yet. Working on it. But toxicity thing is a big link. And you get all kind, again, wasting of muscles, all kinds of problems. Hypercortisolemia, too much cortisol, high blood sugar can lead to diabetes. You ever hear of people taking regular corticosteroid treatments and that causes them to develop diabetes mellitus later on? Cortisol increases blood sugar. So it creates a whole new world of problems. Okay. Reproductive. What we'll do is get a good start on this. I'll hand back these tests, finish up the repro chapter on, what is today? Wednesday? Well, next Wednesday. And then we'll do our second half of the class. We'll do a little review. Okay, so starting with the male reproductive system is a little simpler than the female. Again, the gonads are the sex organs, and the male it's testes. The testes where sperm is made, and testes secrete sex hormones. And then the sex hormones are made in the testes, and then they're going to go through a series of tubes, and eventually these tubes lead to the outside of the body. And what's the first tube that's not listed here that leads into the epididymis? The seminiferous tubules. And then there's a series of tubes that lead from the seminiferous tubules into the epididymis. We're not going to worry about them. But sperm initially enters into the seminiferous tubules. Then from there it's going to go into the epididymis, then the ductus deferens, eventually the ejaculatory ducts, and the final tube leading to the outside would be the urethra. And that's in a male. That would not be the case, obviously. The urethra is totally about urinary function in the females. The urethra in the male is urinary and reproductive functions. And then you have several accessory sex glands. Those are the three major ones there that all contribute to seminal fluid, the fluid that the sperm need to swim in, so to speak, and get nourishment from and protection and all that. So the accessory sex glands are the major producers of seminal fluid. And we'll talk about all three of these a little bit later. And then supporting structures. The scrotum is the sac of skin that the testes would be found within. And I think everybody has at least a ballpark idea of what the penis is. So we'll move on. So the scrotum. And an obvious external divider, it's called the raphe. And the raphe continues deep and just turns into connective tissue, which is the scrotal septum. Show a picture in just a second that divides the scrotum up effectively into two compartments, each one with the testes in it. So real quick, there you have it. There's the raphe would be there, and then it continues on as the scrotal septum, creates two compartments, each one should have a testes. And in the scrotum itself, as well as in the scrotal septum, you have some smooth muscle that's referred to as the dartos muscles. So this is smooth muscle, and it helps constrict the testes for temperature regulation. Sperm need a slightly cooler temperature to develop. So that's why if you ever wondered, that's why the testes are outside of the core of the body. It's because they need a cooler temperature. If you jump into a cold swimming pool, what happens? Excuse me? They shrink. How many people have experienced that here? 1, 2, 3, 4. Hopefully 4, right? Okay. Just checking.
I'm not Lance Armstrong.
All right. Lance Armstrong. Anyway, we'll move on. So you have the dartos muscle that will help constrict the testes, or the scrotum, I should say, and the cremaster muscle, which we'll show you a picture of. A couple of lab models show you the cremaster muscle. It's really bands of skeletal muscle that run through this thing called the spermatic cord that we're going to talk about and attach to the testes. And literally, when they contract, they bring the testes up into the body core for temperature regulation. So there's actually a cremasteric reflex. I don't know how widely it's done, but if you take a reflex hammer and tap the medial proximal thigh of a male, you might activate the cremaster muscle contraction, and you'll see the testes draw upwards. So you can try that at home. Anyway, here's that cremaster muscle. The spermatic cord is just a continuation of connective tissue of the oblique muscles. And it forms a connective tissue sheath that surrounds the vas deferens, a bunch of blood vessels, nerves, and the cremaster muscle. We're not going to get overly anatomically correct here. What is that right there, that ligament there called? The inguinal ligament. And from about there to there, the medial part just above the inguinal ligament, you've got males and females have an inguinal canal. It's literally a canal built into the different abdominal muscles. External obliques, internal obliques, transverse abdominis muscles. So this is the inguinal canal. And the spermatic cord, again, this connective tissue sheath runs through the inguinal canal and then down to the testes. And in the spermatic cord, you're going to have the vas deferens, cremaster muscle, and a bunch of blood vessels and nerves. In females, you don't have a spermatic cord in the inguinal canal. You've got some nerves and some ligaments that help hold the uterus in place. But both males and females literally have like a tube built into the abdominal musculature, so certain reproductive tubes and whatever and blood vessels can pass through.
Is that the same to the testis travels in a 7 month?
Yes. In fact, she mentioned that at about 7 months of age that the the testes are formed near your kidneys, and they're at about that age, due to hormones, they literally descend, they do descend through the inguinal canal. They're supposed to anyway. If they don't, you call it cryptorchidism, which they tend to surgically repair.
Yeah.
And there it is right there. The testes develop in the kidneys in the 7th month, and then they under hormonal influence, they migrate downward, they go through that inguinal canal, and they end up in the scrotum. We're not going to worry about the tunica vaginalis. It's actually peritoneum material. And because the testes are way up here to begin with, they drag some of the digestive peritoneum down with them. So part of the testes are covered by this peritoneum, and they call it the tunica vaginalis. We're not really going to worry about that. And outside on the periphery of the testes, you've got connective tissue that's white colored. They call it the tunica albuginea. Albino, white. So it's just connective tissue. And this connective tissue extends deep into the testes and creates these septum that form 200 to 300 of these little compartments called lobules. And in each of these lobules, you've got a couple seminiferous tubules. So they'll show a picture in just a second, and it's in the seminiferous tubules that spermatogenesis takes place, the development of sperm. So here you see this tunica albuginea here on the outside, just connective tissue, and then it heads inward like that and creates these little compartments where the seminiferous tubules are found. So all these guys are seminiferous tubules. Then eventually the sperm are going to get dumped into the lumen of the seminiferous tubule, pass through all these tubes in here that we're not going to worry about, and enter into the ductus epididymis, where they spend a couple weeks at least, and or hopefully at least, because that's where the site of sperm maturation will be in the ductus epididymis. We'll get there in just a second. So in the testes, all these seminiferous tubules and spermatogenic cells are all the different stages of potential sperm cells. So spermatogonia, primary spermatocytes, etc., etc. Real quick, for the people taking the lab test tomorrow, I'll point and you tell me, probably should have spermatogonia in here, I don't know why. That's spermatogonia are the most primitive. And some spermatogonia will differentiate into primary spermatocytes. But yes?
Would this be the histology slides?
Yes.
Okay,
That's why we're bringing this up.
Yeah, I was just wondering.
So you tell me, and we'll show a picture of a seminiferous tubule cross-section in just a second. Spermatogonia and primary spermatocytes, how many chromosomes?
20. No.
46. They're diploid. You don't begin meiosis 1 until primary spermatocytes turn into secondary spermatocytes. So secondary spermatocytes are the first cells that are haploid with 23 chromosomes. So one primary spermatocyte will form 2 haploid secondary spermatocytes. That's meiosis 1. Then meiosis 2, those 2 secondary spermatocytes will form 4 spermatids that are also haploid. And then the spermatids, at that point, undergo, we'll come to it in a minute, spermiogenesis. They turn into full-fledged sperm. They grow tails. They develop an acrosome with enzymes that can penetrate the egg and all this stuff like that. And then the sperm cells get dumped into the lumen, and eventually from there they're going to go to the ductus epididymis where they mature and learn to swim. Sertoli cells are helper cells for this whole process of spermatogenesis. So we'll list functions of Sertoli cells. They're just regular diploid cells in the testes, and they guide they guide how the hormones work. They literally carry the cells. They literally drag the spermatocytes toward the lumen as they're maturing. They literally release the sperm into the lumen. They provide nutrition. They secrete some hormones. So they're, in a million different ways, helper cells for sperm, future sperm cells. And here's, again, a lot of lists. They're involved in nourishment as the sperm are developing. When a spermatid spermatid just looks like it's a haploid, 23 chromosomes, but it looks like a regular cell. It's going to have to turn into this sperm and grow a flagella and all that kind of stuff. Sertoli cells guide that. So they eat away excess cytoplasm as this sperm is developing from a spermatid. They control all the movements as the cells are maturing. Remember, the least mature cells are on the periphery of the seminiferous tubule. The most mature cells are towards the lumen. And this is all controlled by hormones. They're actually, again, it's called spermiation is the term that's going to come up eventually here, where they release the sperm into the lumen of the seminiferous tubules. They're actually the first source of seminal fluid. The accessory sex glands, like the prostate and seminal vesicles, make the bulk of seminal fluid, but these guys make the earliest contributions to seminal fluid, which helps push the sperm through the early tubes, like the seminiferous tubules and other tubules. They secrete inhibin, which inhibits FSH. It's a mechanism to control sperm development. And we'll get there a little bit when we start talking about testosterone. Testosterone gets released by these Leydig cells. FSH actually triggers the formation of protein called androgen binding protein. That an androgen binding protein will bind to testosterone and keep it in the area. Literally keep it in the testes, and you need testosterone for spermatogenesis. So FSH triggers these Sertoli cells to secrete androgen binding protein. Androgen binding protein binds to testosterone and keeps it in that local area in a high concentration. So you have high testosterone, which you need to make functional sperm. So we'll talk about androgen binding protein again in a second. But FSH triggers the Sertoli cells to secrete androgen binding protein. And that keeps testosterone in high concentrations. Leydig cells in between the seminiferous tubules make testosterone. No testosterone, no spermatogenesis. Ultimately, to make sperm, you need testosterone, FSH, and LH. Any of those ingredients are missing, you can't make viable sperm. And again, seminiferous tubule here, cross section. You've got your outer basement membrane, and then the cells on the outside. What would these guys be out here? Either spermatogonia or primary spermatocytes. So the guys on the periphery are the least developed. All diploid cells. Really, here's their secondary spermatocyte. Now you've undergone meiosis 1. Secondary spermatocytes are now haploid. They're somewhere in the middle. And then here's their spermatid, basically right on the periphery of the lumen. Eventually the Sertoli cells will release them as they develop tails into the lumen, where the little slivers here are actual sperm cells. They're not ready, they need to mature still, but at this point they have tails and they're kind of early immature sperm. Definitely for the lecture in the lab, you want to know this. The male is a lot easier than the female, really. Early on, you got spermatogonia. Some of them are the chosen ones to differentiate into spermatocytes. When does this start happening in a male? At puberty. Hypothalamus, anterior pituitary kind of turn on like a switch. The hormones trigger some of these spermatogonia to differentiate into primary spermatocytes, and then begin meiosis, or what's another name for meiosis? Sexual cell division. Meiosis is the cell division of either eggs or sperm. And the end result of meiosis are what types of cells? Gametes, sex cells, that have haploid number of chromosomes, So either a secondary oocyte in the female, sperm in the male. Primary spermatocytes, right before they begin meiosis, DNA replicates, makes a copy of itself. So you essentially double your genetic material before you begin meiosis. And that allows the end result of meiosis in the male is how many gametes from the starting cell. It would be different for the female. You have one starting cell, we'll start with a spermatogonia, it's up on the board. You're eventually going to get how many sperm from one spermatogonia? 4. Yep. So from one diploid cell, you're getting 4 haploid cells. How is that possible? This one has 46 chromosomes. You have 4 cells with 23, the math doesn't add up. Well, before you begin meiosis, the DNA replicates, makes a copy of itself. They call those chromatids. And basically you have double the genetic material, and that allows you to eventually divvy up all that genetic material into 4 haploid cells. I don't know how much detail you went into meiosis, but big picture, in the male, one starting cell produces 4 gametes, spermatogonia. So again, you duplicate the DNA, then you begin meiosis. Meiosis 1, you make 2 secondary spermatocytes that are haploid. This is where genetic variability takes place. I would put a little asterisk next to crossing over. You double the DNA, and then from there, you can start getting flip-flopping of genes between chromosomes, and that's why crossing over is essentially why everybody doesn't look the same. It provides for genetic variability. Then meiosis 2, you end up with 4 haploid spermatid cells. Then the spermatids, under hormonal development, are going to start developing into sperm. The Sertoli cells help this along, and that's just called spermiogenesis, the genesis of sperm from spermatids. Several words that kind of sound a little bit alike there. So from a spherical cell, you got to develop a flagella. This is the only cells in humans that have flagella. You make a lot of mitochondria, so you have an energy supply, ATP. The Sertoli cells eat away a lot of the excess cytoplasm as the sperm is developing. And then spermiation is just when the Sertoli cells literally release the sperm into the lumen of the seminiferous tubule. They're not really, ultimately the sperm have to become adept swimmers to get to their final destination. And that maturation is going to largely happen in the epididymis. So sperm, when we get there, sperm can 10 days, 14 days, or longer in the epididymis, and that's where they essentially learn to swim and become functional sperm that have the capability to fertilize an egg. And again, this just kind of pretty good chart, but it just puts all that stuff we just talked about together into one little chart. Okay. You make a lot of, males make a lot of sperm. So 300 million is a lot. And sperm cells themselves have a head. That's where the nucleus is going to be with the chromosomes. And then, again, as they're developing into sperm from spermatids, they develop this packet of enzymes that's going to be necessary to penetrate the secondary oocyte and fertilize the egg. So that's what the acrosome is about, is a bunch of enzymes that allow for fertilization to take place. Show a picture of it in just a second. And then the remainder is the tail. You've got the neck at the base of the tail there. I don't know if you remember the organelles called centrioles, but centrioles are responsible they produce microtubules, or proteins called microtubules, that make up cilia, flagella, and in cell division, mitotic spindles. So cilia are just a bunch of proteins, and in this case, that are going to contribute to the actual formation of the tail or flagella. And just two parts of the tail principle versus the very tapering end is called the end piece. It says once ejaculated, most sperm don't live more than 2 days. We're going to find out the egg after ovulation has about a 24-hour window of opportunity. So there's literally a limited window of opportunity for fertilization to take place. They do talk about some sperm can live up to 72 hours, and they refer to them as super sperm. So just throwing that out there. And there's a sperm cell. So there's your enzyme packet. The neck is just a little tapered part, first part. And then you got all the mitochondria for energy production in the middle piece, M for middle, M for mito, and then the tail. And then for the males, females, if you remember, the egg is already partially on the way. You've already started meiosis 1 before you're born. Males, meiosis does not start until puberty. So at puberty, where would this gonadotropin releasing hormone come from? You might want to write that one in there, right here. Where does it come from?
What?
Not the pituitary,
Oh,
hypothalamus. It's one of those releasing hormones. And that controls, again, the releasing hormones and the inhibiting hormones control the anterior pituitary. Yes?
In the sperm, is there like a genetic code already, like when we're swimming, that says it's a male or female?
Well, yeah, now you're getting into the sex chromosomes, the X and Y chromosomes, yes. Yeah. Then, again, hypothalamus turns on and anterior pituitary starts releasing the gonadotropins and spermatogenesis begins. Luteinizing hormone in males, this would be a test question, stimulates the Leydig cells to make testosterone. No testosterone, no spermatogenesis. And the main source of testosterone is the testes. You do make some in the adrenals. And then FSH, again, you need FSH for sperm development, but FSH triggers their Sertoli cells to make this protein called androgen binding protein. And androgen binding protein actually makes testosterone less lipophilic. It makes it less dissolvable in fat, which means essentially that means it can't escape the testes. So androgen binding protein simply keeps testosterone levels high in the testes, and that encourages spermatogenesis. So FSH kind of acts indirectly, keeps testosterone in the area, and testosterone is what directly triggers sperm development. And showing all that good stuff. Anybody ever hear of DHT? Dihydrotestosterone? Testosterone is the most abundant of these two hormones. Some testosterone can get converted into DHT. It kind of reminds me of T3 and T4 from the thyroid. DHT is much, much more powerful, but much less abundant. They both ultimately have masculinizing effects. DHT is the one that gets implicated in male pattern baldness, is one example. But it's a more powerful version of testosterone, but in lesser amounts. And collectively, they're both involved in the secondary sexual characteristics, all the various changes that you see at puberty. And, libido, sexual drive in males. Low testosterone, low sexual drive. Not to go on tangents, estrogen to testosterone ratios in males, way out of whack in this modern day and age. They're finding, when they do blood work on males, they're finding really high estrogen levels and really low testosterone levels. And if you're a male, that's not what you want. Real bad. All the chemicals in the environment are triggering massive estrogenic effects in males, and definitely leads to negative effects. And